Welcome to this explainer. Today, we're diving into some advanced bio. We are going to bridge that microscopic world of cellular division right up to the macroscopic traits we actually inherit. If you've ever wondered about the hidden mechanisms making us exactly who we are, well, you are in the right place. We're connecting the tiny blueprints inside our cells all the way up to the observable traits we share with our families. Let's get right into it.
So why do you look like your parents, but not exactly like them? We're starting off with this classic biological puzzle of genetic diversity. It perfectly sets the stage to see how a very specific type of cell division, called meiosis, solves this exact problem. It's literally nature's ultimate balancing act, passing down the instructions for life while ensuring every single generation is uniquely its own.
Here is our roadmap for the explainer today. First, DNA and your chromosomes. 2. Meiosis, the Great Divide. 3. Creating genetic variation. 4. Mendel and basic heredity. And finally, 5. Predicting traits with Punnett squares.
All right, Section 1, DNA and your chromosomes.
Let's start with the blueprint itself. We need to look at how these incredibly long strands of DNA are packaged with proteins into distinct structures called chromosomes. Because to really grasp heredity, we have to see how this genetic material is organized. In a human cell, we see 23 pairs of chromosomes. Now, what's crucial here is that these pairs are homologous. That means they're similar in length, similar in appearance, and they carry the exact same genes, but the two copies might differ in the specific details. You get one chromosome from your mother and one from your father to make up each and every one of these 23 pairs. Looking at a real human karyotype, it's pretty wild to see. You'll notice those first 22 pairs are what we call autosomes. These hold the genes for general characteristics that aren't directly related to your sex. Then, just like at that final pair at the bottom right, the specific sex chromosomes. These directly control sexual development. 2 X chromosomes mean female, while an X and a Y mean male.
Moving right along to Section 2, Meiosis, the Great Divide.
To understand how we actually reduce our chromosome number by half to create gametes, the sperm and egg cells, we need to look at the first 4 distinct phases of meiosis 1. Keep in mind, meiosis as a whole has 2 main stages, but this first half, this is where the magic of sorting our homologous chromosomes really happens. We've got prophase 1, metaphase 1, anaphase 1, and telophase 1. Starting with prophase I, the DNA coils up tight, and the nuclear membrane begins to disappear. But here is the absolutely critical event. Those homologous chromosomes pair up in a process called synapsis to form what's known as a tetrad. You can just think of a tetrad as a foursome made by 2 chromosomes locking together. As we slide into metaphase 1, notice how the chromosomes align perfectly at the equator, the dead center of the cell. Meanwhile, out at the edges, you've got these green structures, the centrosomes. They're using spindle fibers to reach out and grab the chromosomes right at their center point, which we call the centromere. So the crucial turning point hits in anaphase 1. This is the physical separation. The chromatids get attracted and actively pulled to the opposite poles of the cell. The pairs are literally being divided, making sure that the resulting cells are going to end up with exactly half the original number of chromosomes. And telophase 1 wraps up this first half of the process. The spindle fibers disassemble, and a new nuclear envelope forms around those newly created daughter cells. These cells are now totally primed for meiosis 2, which will ultimately give us 4 haploid daughter cells. But, what happens to those cells next depends heavily on whether we're looking at male or female biology. Looking at gametogenesis, which is just the process of these cells maturing, it's absolutely fascinating to contrast the 2 different pathways. Spermatogenesis is all about mass production. We're talking 4 active sperm cells with tightly packed DNA and a whip-like flagellum for movement. Oogenesis, on the other hand, is a totally different strategy. It prioritizes quality over quantity, investing all its resources into creating just one single mature egg. The rest of the genetic material, it just gets discarded as unused polar bodies. You can really trace this dramatic difference in resource allocation. If you follow the primary spermatocyte dividing equally on one side, it goes all the way down to 4 functional sperm. But trace the primary oocyte, and you see this highly asymmetrical division. That's what leads to that one single, incredibly resource-rich ovum on the bottom right.
All right, let's hit Section 3, creating genetic variation.
So exactly how does meiosis actively increase genetic diversity? I mean, this is the fundamental biological answer to why siblings look different from one another, even though they have the exact same parents. Family genetics can honestly feel like a mixed bag of candy, and this gummy bear tree perfectly captures how combinations of traits get passed down and shuffled around. Through crossing over in prophase 1, where chromosomes literally swap chunks of DNA, and independent assortment, where they sort themselves completely randomly, the genetic deck is thoroughly shuffled before passing anything on to the next generation.
Let's zoom out a bit for Section 4, Mendel and Basic Heredity.
Moving away from the microscopic world for a minute, Gregor Mendel actually figured out the rules of how these genes operate way before anyone could even look through a microscope and see a chromosome. To really grasp his laws, we first have to establish the stark difference between dominant and recessive traits. Dominant traits, which we represent with capital letters, are the more present characters. They can be homozygous, pure, like 2 capital letters, or heterozygous hybrid, meaning 1 capital and 1 lowercase. Recessive traits, though, those are the less present characters, and they must always be pure, meaning 2 lowercase letters, to actually show up physically.
Which brings us to our final part, Section 5, Predicting Traits with Punnett Squares.
We can actually use a mathematical diagram called a Punnett square to predict breeding outcomes. That physical separation of chromosomes we just saw in meiosis, it translates perfectly onto this grid. Mendel's first law, the law of dominance, reveals this super consistent 3-to-1 ratio of dominant to recessive traits when you cross 2 heterozygous parents. This was a massive breakthrough, folks, because it proved traits don't just blend together into some muddy middle. They're distinct, particulate elements.
Looking at a monohybrid cross in the second generation, you can literally see this math in action. By crossing 2 hybrid purple pea plants, the physical separation of those homologous chromosomes during gamete formation results in exactly 75% purple-flowered plants, the dominant trait, and 25% white-flowered plants, the pure recessive trait. Boom! There is our 3-to-1 ratio in action.
But what happens when things get, slightly more complicated? When we expand this out to a dihybrid cross involving 2 differing pairs of characters, Mendel's second law, the law of independent assortment, kicks in, and it reveals this beautifully predictable 9-to-3 to 3-to-1 ratio in the second generation. By tracking 2 traits at once, like pea color and pea shape, this extensive Punnett square proves that these genetic elements sort entirely independently of one another. Getting a yellow pea absolutely doesn't mean you have to get a round pea. Out of the 16 possible combinations, you get 9 yellow round, 3 green round, 3 yellow wrinkled, and 1 green wrinkled. It is biological math at its absolute finest. After seeing how these microscopic mechanisms, from homologous chromosomes pairing up in a tetrad to crossing over to the incredibly predictable laws of dominance, after seeing how they drive the vast mathematical scale of heredity, well, it's pretty humbling. Because independent assortment alone creates literally millions of possible genetic combinations in human gametes. Add crossing over to that, the numbers become almost infinite. So I'll leave you with this thought to marvel at. Given the staggering, astronomical biological lottery required to sort and pair those specific traits, have you truly grasped just how spectacularly unique you are?